Tissue engineering focuses on the development of devices capable to restore and maintain normal function in diseased or injured tissues. Most of the native tissues are composed by different types of cells and extracellular matrices in specific spatial hierarchies. For example, articular cartilage consists in different zones with varying types and orientation of collagen fibers and collagen-binding proteins. The main strategies adopted in tissue engineering can be divided in monophasic, biphasic and triphasic approaches. Biphasic and triphasic approaches use 2 or 3 different pore architectures, materials or fillers to prepare multi-layered functional devices. A single material can also be used to achieve biphasic or triphasic devices if it is possible to create a gradient of 1 or more physical properties. In the frame of this work, a fast and simple route to achieve polylactic acid-based 3-layered pore scaffold is presented. Differently from many other scaffold production technologies, the strategy here adopted can be considered eco-friendly since it does not require any toxic solvent potentially dangerous for environment and for living cells and tissues. In particular, the pathway proposed combines melt mixing, compression molding and salt leaching. Both the morphological analysis and the mechanical behavior of the structures were investigated, paying particular attention to the processing structure properties relationships of the prepared devices. Sodium chloride was ground in a laboratory blender for 20 minutes. Thereafter, sodium chloride was dried on a heater at 100 degrees C prior to sieving in a sieving machine.